Welcome to Click for IB Biology. We're in topic D 3.1, and now we're looking at the process of fertilization in plants, the formation of seeds, and their germination. There's a blank PDF of this presentation that's linked in down in the description. Download that, annotate with your own notes as we go along, and then why not take the 10-question quiz, which is at the end, also linked down in the description. Answers are there, and you can use that to evaluate your own learning. So in the previous videos on pollination, we described how pollen grains from the anther, how these would be transferred to the stigma. And in particular, we were describing insect pollination, but the presentation here is also consistent with that of wind pollination. So the anther would land on the stigma, and the stigma surface would contain water and perhaps even some sugars. And this would allow what we would call the germination of the pollen grain, a slightly confusing term, in which it would begin to grow a tube that will grow down through the style. Now at this point we can come back to the idea of preventing self-pollination or hybridization, because at this point there's interaction between the pollen grain and the stigma and style cells in what is known as SI, or self-incompatibility, in which the pollen grain is not allowed to grow because of interaction with the cells of the stigma. So this involves receptors on the surface, and it prevents the growth of the pollen grain. However, let's assume that we have successful cross-pollination, and the tube will begin to grow down through the style towards the ovary, and it will move in and into the ovary space like so. Now let's consider the pollen grain for a moment. The pollen grain actually contains two haploid nuclei from the process of meiosis, which took place in the anther. And the pollen grain is attempting to locate the plant eggs, which we would know as ovules. And we'll look at the details of this process in just a moment. So the pollen tube will grow down and towards the ovule, and will enter the ovule through a hole which is called the micropyle. At that point, the nuclei, which have been progressing down through the pollen tube towards the ovule, then they can move towards and carry out the process of fertilization. Okay, so let's take a look at the actual fertilization itself. So here we have my pollen grain with its two haploid nuclei, and then here's the ovule. Now curiously, the ovule actually has three haploid nuclei, one in this location here and two further up here. There's the hole in the ovule structure, which is the micropyle, and the pollen tube will grow down and locate the micropyle like so. And then these nuclei are going to move down through the pollen tube and then come into the ovule itself. One of those male pollen grains will fertilize, will combine with two of the nuclei, and they're going to form a triploid, which is an unusual idea, a triploid structure called the endosperm. And these cells, which result from the mitosis of this triploid cell, will form the food store. The other nuclei will join with this single ovule nuclei here like so, and that fertilization is going to give rise to what we would recognize as being the embryo, the new plant. So we actually have in plants what is called a double fertilization. These details help us to understand that when we look at the structure of a seed, the outside of a seed, or what a seed is, has a seed coat, this structure called the test around the outside, and within it are the embryo. The embryonic shoot is called the plumule, and the embryonic root is called the radicle. So these together are the embryo resulted from that fertilization that created the diploid structure. The food stores in this location here are inside this seed coat, and the seed coat protects the seed as it's exposed to the environment and prior to germination. So if we take a brief look at the concept of germination, germination requires essentially two features, one of those being water and the other being temperature. Specific types of seed require additional features, but the basic idea is the need for water and very particularly temperature. And with the temperature, of course, this is linked to optimal enzyme function levels, and the water allows the nutrients to create into solutions. So within the seed, what happens is that the outer seed coat is responsible for the production through the expression of genes of plant growth regulators or plant hormones called gibberellins, and in turn the gibberellins result in the production of amylases, and the amylases break down the starch which are stored in the seed into maltose, and of course that maltose is then taken up by the embryo to support its respiration with the further breakdown to glucose, and we can begin aerobic respiration. The water necessary to create these solutions enters through the micropyle, or indeed through the seed coat, which perhaps by this time has already been abraded and worn down. And the embryo begins to grow the embryonic shoot, which will grow upwards towards the surface. And of course, the embryonic root will grow downwards in a tropic response down into the soil, where it will be able to harvest further nitrates and phosphates to support the growth of the embryo. So that's a simple outline of the process of germination. In the next video, we're going to go on and consider how the seeds might be dispersed to reduce competition with the parental plant. That's the video linked on the left. A chance to subscribe to the channel, it really encourages me if you do that. And on the right hand side are the playlists with all the videos summarized in D3.1 reproduction. Success with your studies.